Okay, so in the previous video we had finished the basal domain of the epithelium. So we said that, okay, this is an epithelial cell, and this right here was its basal domain, right? The basal surface. And at the basal domain we said it has a basal lamina right outside, and then there's a reticular lamina, and all of this is the basement membrane. This is the basal lamina again, and this is the reticular lamina, and then we might have some connective tissue here connecting this epithelium to some other tissue below, maybe some muscle here or something, whatever. And the basal surface contained hemidesmosomes, right? We'll study the details of hemidesmosomes in this lecture as well. But what we're going to focus on today is the lateral surfaces of epithelia, right? And because we said that epithelium or epithelial cells are actually aggregated cells, they are collected, tied up cells, then they should have some junctions connecting them to other cells, right? And that's exactly what they have, is that they have some junctions, adhesions that connect epithelial cells together, and that's what we're gonna be studying today. Now, most of these junctions lie on the lateral domain or the lateral side of the epithelial cells, yeah. So this would be the lateral side, and there's junctions here that are connecting these cells together. But there's also this one junction that we already studied at the basal surface, and this is called a Hemidesmosome. Okay, so let's go ahead and try to look at this little diagram that I got from the Junqueira's book of Histology. I have a link for it in the description if you want to buy it. It's a very good read. It's a wonderful book if you want to build up histology concepts. And we have permission to use these diagrams, and so here it is. So let's assume this entire thing is an epithelial cell. Okay, this is taken from an intestinal columnar epithelium. So we have some microvilli on top. We'll study what those are in the next video. But basically, because right now we're studying the lateral modifications, we've already done with the basal modifications. Once we do the apical modifications, the first one will be the microvilli, and I'll tell you what those are. Basically, those are little projections that are able to increase surface area for absorption of stuff, right? Absorption of nutrients and whatnot. But that's not our focus today. Our focus is on the lateral domain, and so what's present on the lateral domain are some junctions. Firstly, we have the topmost junction. Let's start with this one. Let's call it number 1. Junction number 1, which is present at the most apical site of an epithelial cell on the lateral domain, is the tight junction. So tight junction would be the closest to the apex of a cell. So from the apical domain down towards the basal domain, the first junction that we see is the tight junction, which is present at which is the nearmost junction to the apical surface, and that is the tight junction. Now, tight junction, which from its name you should be able to understand that this is a very, very tight sort of linkage between cells, and it is present at the nearmost point of the apical surface. So this is a very tight junction, and what it does is that it basically seals off the gaps between the two cells so that nothing can come in the intracellular space right here. So there's a space right here, and that space is closed off by this tight junction because it's really, really tight, and it's closest to this sort of basal surface that is present there of the cell. So basically what it does is, apart from linkage, it basically closes the intracellular gap and prevents stuff from flowing into intracellular spaces between the cells. And a tight junction is made up of, obviously, proteins, and one of the proteins that is made up of are called occludins, and another one is called claudin. Claudin proteins or claudins, and because of this protein, this junction is also referred to as zonula occludens. Okay, so tight junctions or zonula occludens or occluding junctions are made up of occludin proteins and claudins, and what they do is that they help bridge two cells in a very tight junction to close off that intracellular gap to prevent stuff from flowing into and between the two cells, and it is often found in places where barriers are present. So, for example, there's the blood-brain barrier, and tight junctions are really important in blood-brain barrier. When we'll go to the nervous system chapter, I'll show you a diagram of it. Similarly, there's also in the testes, there's something called blood-testes barrier, and those also contain tight junctions. Tight junctions are also found in the intestinal epithelium. Where they prevent the stuff that is present inside the intestinal lumen from leaking into your body tissues. So that's mainly their function, is that they prevent stuff from escaping through the intracellular spaces. Now there's a little detail to it, is that a tight junction, just like any other cell, there's cytoskeleton of the cell, and cytoskeleton is made up of many different kinds of filaments. Some of them are called actin filaments and intermediate filaments and so on. Tight junctions are actually connected on the inside by actin filaments, okay? And also these junctions span the circumference of the cell. As you can see in this diagram, they span the cell. And that's one of the features of tight junction that make them slightly distinguished from junctions like desmosomes and so on. So on the inside, they're connected to actin filaments. Also down in neat notes that you can get at our website, and they span the cell, and so on. The examples that I mentioned were the blood-brain barrier and blood-testes barrier and so on. This is made up of cerulite cells. This is made up of endothelial cells and astrocytes and so on. That you'll study later. Then next up we have number 2 in the series. Yeah. So after the tight junctions, the nearmost junctions near the apical surface would be the adherence junctions, or also called zonula adherens, right? Just like the tight junctions or occluding junctions are called zonula occludens, similarly adherence junctions are also called zonula adherens. So let's write that down. Zonula adherens. And basically, the function that they perform is that they help anchor, they help adhere, they help in cell-to-cell linkage and stability. So what they do is that they glue the cells together. They provide a very strong bond between two cells for linkage, for stability, for support, so that the cells cannot escape that epithelium, and they're closely tied to the other cells and basically the tissue. And so you'd assume that if there was a problem in the adherence junctions, then you'd expect the cells to escape that epithelium, right, to be free, because there's no more linkage and stability that is brought about by this anchoring junction that is really, really strong. And zonula adherens is actually made up of what's called cadherin proteins. That's a family of proteins, very important for cancers. Research students as well, and we have catenin or catenin proteins, whatever. There's one called E-cadherin, very important, and if there's a problem with these, then you'd expect the cell to escape that particular tissue. And that's called epithelial to mesenchymal transition. I'll explain that to you in just a bit, but before that, let me just quickly mention the fact that adherence junctions, which are zonula adherens, which are made up of cadherin and catenin proteins, they also span the cell, right? They span the circumference of the cells. They would be present 360 degrees all around the cell, and they are also connected to actin filaments. Okay, so they're also connected to actin filaments, and they also span the cell. So let's quickly write that down before I explain to you what epithelial to mesenchymal transition is. So connected to actin filaments and span the cell. Now, epithelial to mesenchymal transition, or the EMT pathway, is something like this. I'll quickly zoom this in. So we have a set of epithelium, right? And the cells in the epithelia escape off, and they become mesenchymal, which would mean they would not be aggregated anymore. They would not be bound by anchoring junctions or linkage and stability junctions that link them together and hold them in place. So if those junctions are gone or damaged, especially happens in some cancerous cells when they start to basically migrate and basically become what's called metastasis. When they start metastasizing or spreading, then this basically junctions are lost, and they undergo this epithelial to mesenchymal transition, in which those cells would leave off the epithelial, their epithelial nature, and the junctions would be broken, and they would be then free cells, and they would be able to basically migrate towards different places and harm those places, and maybe develop cancerous tumors at those places as well. And that process is called metastasis. And this is the epithelial to mesenchymal transition. Now let's come back to what we were doing, and the next junction in the series is actually called a desmosome. Well, we studied the hemidesmosomes, now we're studying the desmosomes. So that's number 3 in the series, which is just below the adherence junctions with respect to the apical surface. And let me actually make some space here and erase some of the stuff. Now, desmosomes are another type of anchoring junctions, which would mean that, just like the adherence junctions, they would provide linkage and stability. They would hold two cells in place, and they would connect the two cells in a very strong bond so that they're not able to escape. They remain part of that tissue, and they have this linkage and stability and anchoring point, right? And that's the desmosome as well. So desmosome is also another kind of anchoring junction. And desmosome, just like adherence junction was called zonular adherence, desmosome is actually called macula adherence. So write that down here, number 3 in the series. Another word for desmosomes are macula adherents. And again, I want to mention here is that all of these junctions are present on the which domain? The lateral domain of the epithelial cells. And desmosomes, again, is also the proteins that form desmosomes are also some cadherin family proteins. So we have some cadherins here. And we have very important ones, desmoglein, and this one right here, desmocolin. Good to remember in case you face some pathologies explaining these two proteins or in the future. Now, the difference between desmosomes and adherence junctions, apart from obviously they're further away from the apical surface, is that desmosomes do not span the cell. You see these adherence junctions right here? They are spanning the cell, right? Spanning 360 the cell. Desmosomes are actually present at their own specific locations, don't span the cell. And another thing is that they are connected to intermediate filaments and not actin filaments on the inside, right? So these are the two differences between adherence junctions and desmosomes, is that, first of all, they don't span the cell. And secondly, what was it? They are connected to which filaments? Intermediate filaments. Okay. And the function that they provided, again, was the anchoring function, the linkage and stability of two cells together. Next up in the series is a very high yield junction. It is my personal favorite out of all of these. It is called the gap junction, also called the communicating junction, because it allows actually communication between two cells. It allows transmission of or transport of materials from one cell to another between neighboring cells. And the cells are able to transport materials, they're able to communicate with each other, and so on. In some cases, these cells can actually even transmit electrical impulses through these gap junctions. And the way to do that, an example of this is the cardiac muscle. We'll study the details of that when we do the cardiac muscle, and you learn that in cardiac muscle physiology as well. But yeah, a good example of where the gap junction would be present is the cardiac muscle cells. The gap junctions are made up of some channel proteins called connexons, or connexins that join to form connexons. So we have connexin proteins. Actually, I should write this down. Connexins are actually joining together to form these connexons. and those are present inside the gap junctions, and they form these channels that are able to transport materials, information, ions, signaling factors, and so on, depending on the cells where they are present, to the other cells. And so the cells are able to communicate with each other, and this is why it's called the communicating junction. Some of the examples where they're present is embryo mesenchyme, right? In embryo, developing embryo, you don't have a nervous system, you need cell-to-cell communication. So in embryo mesenchymal cells, you have gap junctions. Similarly, you have one of the most common examples is cardiac myocytes. Yeah. And another one is osteocytes. Who knows what osteocytes are? Anyone know what osteocytes are? Any guesses? These are the cells of your bone, the mature inactive cells of your hard bone matrix. And I actually teach the details of osteocytes and everything, even the gap junctions that are present there, in a chapter that's coming up shortly in this series. One thing I want to mention here is that I won't be displaying any slide images of the junctions, because I will show those images once we discuss the chapters themselves, so that the only images that you see are the ones that you need to remember. So for example, in osteocytes, you'll see some gap junctions images in their own little canals. Similarly, in cardiac myocytes, you'll see some gap junction images as well, because you need to remember those, right? Otherwise, showing you random examples in the start of the series would be useless and inefficient. Similarly, I would also show some desmosomes that are present inside skin cells, right, when we do the skin cells chapter. But right now I'm not going to put any examples of these junctions, because you don't need to remember that many junctions in medical histology with regards to microscopic images. And last but not the least, we have the hemidesmosome, right? We're finally at the hemidesmosome. This is again another type of anchoring junction. You already know that it helps cell binding with the basal lamina. It helps the cell to actually adhere to the basal lamina for linkage, support, stability, for protection as well. It's there. And this hemidesmosome, we said it was made up of proteins called integrins, right? Integrins. And it was bound outside to a protein inside the basal lamina called the laminin, right? Laminin, and so on. And they were bound to each other, forming this hemidesmosome. Now, a clinical correlate to this would be the beta integrin. We have beta integrin, right? Beta integrin is obviously an integrin molecule present inside hemidesmosomes in the skin. And what happens is that there's a recessive mutation of beta integrins, which results in this pathology or skin blistering disorder called epidermolysis bullosa. Epidermolysis bullosa, which basically means epiderm would be the skin, and lysis, which would mean breakage. So obviously there's breakage in the skin epithelium, which is resulting from a mutation in beta integrin. So beta integrin is mutated, and so the hemidesmosome is not able to function correctly, and then there's epidermolysis, which means breakage of the skin, and it results in these blisters or bullosa, right? And explain this in the first video as well. I think that we have a layer of cells and they're bound to a basal lamina, but if these hemidesmosomes are mutated or they're damaged, then what would happen is some of these cells would come off from the basal lamina, and then there would be a potential space created here, and water would move from the tissue fluid into this particular space. We still have many different layers of epithelium here, and so on. And this would cause a blister. Right, so let's go ahead and have a peek at our neat notes that I have compiled for you here. You can take a screenshot of them if you want, but they contain the information that we learned in this video. And I remind you that these are integrated notes. They don't have they don't go as a separate atlas, but they are actually integrated with the videos. So it's good. They go with the videos. So you'll have to see the video first and then read the notes, and you'll be able to understand everything and so on. Secondly, I want to state here is that this diagram is taken from Junqueira's book of Histology. We have permission to use these diagrams, and that book is a recommended book. I recommend it personally as well. It's a wonderful book. It is a very good read, and if you like reading books for comprehension of different concepts, then I absolutely recommend it. And we actually have a link in the description. If you plan on buying that book, then please use that link. And yeah, good luck. I'll see you guys in the next video.